and welcome to my channel. So today we are going to discuss about the renal tubular processing. It's the fifth lecture of the series, and we have plenty discussed about the ascending limb of loop of Henle, and now we will discuss about the early distal tubule. As you know that the nephron consists of the proximal convoluted tubule, the descending limb of loop of Henle, the thin part of ascending limb of loop of Henle, then the thick part of ascending limb of loop of Henle, then the early distal tubule, the late distal tubule, cortical collecting tubule, and the medullary collecting tubules. So let's discuss about the early distal tubule. We are discussing the part present here, right? Okay, we are focusing on this part. This is the early distal convoluted tubule. At the junction of the ascending limb of loop of Henle and the early distal tubule, there is the group of cells known as the macula densa. Okay, there is a group of cells macula densa, and these cells are sensitive to the concentration of sodium and chloride reaching here. Sodium and chloride reaching the macula densa. What does it mean? It means that if sodium and chloride concentration is high, it means that the glomerular filtration rate is high. Why so? It is because that the GFR when is high, the speed of the tubular fluid increases and the proximal convoluted tubule cells find less time to reabsorb sodium and chloride. So sodium chloride concentration increases, and when it reaches the macula densa, so macula densa identify that GFR is high. It stimulates the juxtaglomerular apparatus to cause vasoconstriction, the afferent arterioconstriction. And when the sodium chloride concentration reaching the macula densa is low, it means that the GFR is low. GFR is lower than 120, because when GFR is low, the speed of the tubular fluid is slowed down, and proximal convoluted tubule cells find the time to reabsorb sodium and chloride, right? This makes sense. And when GFR is low, the macula densa signals to the juxtaglomerular apparatus to cause vasodilation, so that more plasma flow can occur from the blood, and the GFR can be increased in that way. Having discussed that, let's talk about the early distal tubule. The early distal tubules share features with the ascending limb of loop of Henle. They both are impermeable to water. They do not allow free reabsorption, or they are not freely permeable to water, and these both paths pump out solutes from the luminal fluid. It means that they make the luminal fluid diluted and the interstitium concentrated. So the ascending limb of loop of Henle and the distal tubule causes the interstitium to become concentrated or hyper or smaller. Okay, and the early distal tubule is also known as the cortical diluting segment, because it is diluting the tubular fluid. It is diluting the tubular fluid present in the lumen of the nephron. So having said that, so let's discuss about a typical cell of the early distal tubule. First of all, you need to understand that these cells have a special pump known as the sodium potassium ATPase. Under the influence of ATP, they pump sodium out and potassium into the cell against the concentration gradients. Okay, and when the sodium potassium ATPase pumps the sodium out, it causes the cell to be sodium poor. Okay, and there comes the function of the second transporter known as the sodium chloride co-transporter present on the luminal membrane. This pump, okay, because it is present on the luminal membrane, as the cell was poor in sodium, this pump allows the sodium to move into the cell along with the chlorine, okay, along with the chloride ion, and this is known as the sodium chloride co-transporter. One of the most important thing you need to understand that in case of the ascending limb of loop of Henle, there is a special pump known as the sodium potassium 2 chloride co-transporter. This is reabsorbing sodium as well as the potassium as well as the chloride. Okay, these are reabsorbing all of these solutes into the cell, and these solutes can move out from the cell via special mechanisms. When GFR is filtered, the tubular fluid in the PCT, it causes 65% of the sodium reabsorption in the PCT, and in the ascending limb of loop of Henle, about 25% of the sodium reabsorption is occurring here. So 65 plus 25 is 90%. Okay. So when the tubular fluid reaches the earlier part of the distal tubule, approximately 90% of sodium has already been reabsorbed, right? 90% of the sodium has already been reabsorbed, only 10% of sodium is left, and from 10% approximately 5 to 8% will be reabsorbed in the distal convoluted tubule, right? So I want to put in your mind one thing, that there are two kinds of drugs that are used as far as the nephron is concerned. First is the loop diuretics, we have discussed in the previous lectures, and second is the thiazide diuretic drugs. So the loop diuretics will inhibit the sodium potassium 2 chloride co-transporter. The loop diuretic will inhibit this pump. So will the sodium reabsorption occur? No. Will the potassium reabsorption occur? No. Will the chloride reabsorption occur? No. So all these solutes, the sodium, potassium and chloride, will move down the drain into the urine when we offer the patient loop diuretics. When the patient is ingesting loop diuretics, he will be suffering from natriuria. Natriuria means that lot of sodium is present in the urine, as sodium is a solvent attractant. It means that it attracts the water, so the loop diuretic will promote the diuresis. That's why these drugs are known as diuretics. So lot of water will be drained out along with the sodium. When we talking about loop diuretics, they work in the ascending thick part of the loop of Henle. But when talk about the thiazide diuretics, they do not work here. They work in the distal tubule, the distal convoluted tubule, especially on the sodium chloride co-transporter. And as expected, this thiazide diuretic will block the sodium chloride co-transporter. And when the sodium chloride co-transporter is blocked, can sodium be reabsorbed here? No, sodium cannot be reabsorbed here, and sodium will move down the drain. But the most important thing to mention here, that amount of sodium reabsorption here and amount of sodium reabsorption here is different. In the ascending part, 25% of sodium is reabsorbed, and in the distal part only 5 to 8% of sodium is reabsorbed. It means that loop diuretics will promote more diuresis because they are responsible to reabsorb 25% of the sodium. Okay, and thiazide will produce comparatively low diuresis and low natremia. Okay, natriuria as far as the sodium is concerned. The same thing is written here, that in case of loop diuretics, they can influence more diuresis because they will inhibit 25% of sodium reabsorption. But as compared to thiazide, they will influence less diuresis. They promote less diuresis because they will inhibit only 5 to 8% of sodium reabsorption. We have discussed the sodium reabsorption in the distal tubule. Let's revise it. There is pump sodium potassium ATPase on the basolateral surface, and there is a pump sodium chloride on the luminal surface or membrane. The sodium potassium ATPase will constantly pump the sodium out and potassium in, and the sodium chloride, they have the concentration gradient because the sodium is less inside the cell. So sodium will move from lumen into the cell, with the help of sodium chloride co-transporter. Okay, I hope you understand this, like sodium reabsorption. Let's talk about the calcium reabsorption in the early distal tubule. So, first of all, the calcium reabsorption in the early distal tubule is transcellular. It means that it is occurring through the cells. In case of ascending limb of loop of Henle, the calcium reabsorption was occurring in between the cells. It was paracellular reabsorption. But in case of the earlier distal tubule, the calcium reabsorption is paracellular. No, not paracellular, it's transcellular. Transcellular means that the calcium reabsorption is occurring through the cells, okay? And the second thing, the influence of the parathyroid hormone on the calcium reabsorption is very important because there is a receptor for the parathyroid hormone, the serpentine receptor for the parathyroid hormone. It is present on the basolateral surface. Okay, let me erase some of the things so that you can understand more easily. First of all, this is the receptor for the parathyroid hormone present on the basolateral surface. And when the parathyroid hormone attaches to this receptor, it is connected to the G stimulatory protein. This G stimulatory will stimulate the adenylyl cyclase. Okay, adenylyl cyclase will convert ATP to CAMP, the cyclic adenosine monophosphate. This CAMP will promote the production of protein kinase A, and this protein kinase A will phosphorylate many proteins that will increase the calcium reabsorption. Okay, that will increase the calcium reabsorption. Same thing is written here, that receptor for the parathyroid hormone is present on the basolateral surface. G stimulatory protein will stimulate adenylyl cyclase CAMP, then protein kinase A. The protein kinase A will phosphorylate many proteins that increases the calcium reabsorption. Okay, there are two more calcium transporters that are present on the basolateral membrane. Let's discuss about this. First of all, the calcium ATPases. This is the calcium ATPases number 3. Under the influence of ATP, they will constantly throw the calcium out of the cell. They are constantly throwing the calcium out of the cell. And the second transporter for calcium is calcium sodium exchanger. It's calcium sodium exchanger. And what will this do? It will cause the calcium to move out in the exchange of sodium to move in. It will cause the sodium to move in and calcium to move out. So these two transporters are constantly throwing the calcium out of the cell. Okay, and they causes the cell to become calcium poor. Okay, they causes the cell to become calcium poor. And when the parathyroid hormone causes the phosphorylation of these two exchangers, the calcium ATPase and the calcium sodium exchanger, so they cause phosphorylation of these two exchangers. And when the cell became calcium poor, so from the lumen it became easier for the calcium to diffuse into the cell, because the cell is already low poor in calcium. So from the lumen, calcium can move into the cell very easily. Okay, it's understandable. Talking about the thiazide diuretics, how the thiazide diuretics will influence the calcium reabsorption. We have discussed that thiazide diuretic will inhibit the sodium reabsorption and they will produce mild diuresis and natriuresis. But the loop diuretics will produce even severe diuresis and severe natriuresis as compared to the thiazide diuretics. So, it means that thiazide diuretics are influencing the calcium reabsorption as well. How does it work? Let's see about this. Thiazide diuretics increases the absorption of calcium. It's very important to understand it. Okay, But as compared to the loop diuretics was inhibiting the calcium reabsorption and magnesium reabsorption. We have discussed how in the previous lectures. Talking about the thiazide diuretics, thiazide will inhibit the sodium chloride co-transporter. It will inhibit what thing? It will inhibit the sodium chloride co-transporter. Can sodium move in? No, because this transporter has been inhibited by the thiazide. So sodium cannot move in, and the the sodium inside the cell became low. Okay, but the cell cannot tolerate this. It will start to exchange sodium with calcium. Through what system? This system, calcium sodium exchanger. When thiazide blocked the sodium chloride co-transporter, the cell became sodium poor. Okay, so it starts to exchange sodium from calcium. It will bring the sodium in and it will throw the calcium out constantly. So what does it mean? It means that the cell now starts to become hypocalcic. It means that the cell became poor in calcium. This make a concentration gradient for calcium, and calcium will move from lumen into the cell. So as a conclusion, what we can say? We can say that thiazide diuretics are hypocalciuric drugs. Hypocalciuric drug, it means that they do not allow the excretion of calcium into the urine. But as compared to loop diuretics are hypercalciuric drugs. They promote the like loss and the drainage of calcium into the urine. They don't care. But as compared to thiazide diuretics are hypocalciuric drugs. Let's discuss again. It's worth repeating. The thiazide will inhibit the sodium chloride cotransporter. Can sodium move in? No, it cannot. So, cell becomes sodium poor. It starts to exchange sodium with calcium, sodium moving in and calcium moving out. When calcium moves out of the cell becomes poor in calcium, and calcium from the lumen starts to move inwards along the concentration gradient. Okay, that's why the thiazides are hypocalciuric drugs. In the patients, in some families there is some kind of defects known as the calcium handling defects. It means that those families or the person of those families are always in hypercalciuric state. They are always losing high amount of calcium into the urine. Is it a good condition? No, it's not, because when calcium passes through the nephron, it increases the risk for the formation of the kidney stones, urolithiasis, nephrolithiasis. The conditions, the risk for urolithiasis and nephrolithiasis starts to increase. Is it a good news? No, it is not. So, in those patients or in those families, we can give thiazide mild dose. The mild dose of thiazide will reduce the risk for the kidney stones. Okay, I hope you understand the earlier part of the distal tubule. Let's talk about the late part of the distal tubule, this cell. The late part of the distal tubule and the cortical collecting tubule. This is the cortical collecting tubule. We are not discussing about medullary collecting tubule, only the cortical collecting tubule. They both have same functional characteristics. And the type of cells here are two kinds. First is the intercalated cells and second is the principal cells. Principal cells are more abundant and intercalated cells are present in between the principal cells. So first let's discuss about the intercalated cells. The intercalated cells are nothing but proton secreting cells. They are constantly pushing the proton into the lumen. They are proton secreting cells. If you want to remember one thing about intercalated cell, remember this, that they are proton secreting cells. How come? Let's discuss about this. Inside the cell, of course, all of the cells have water inside them and carbon dioxide inside them, right? So carbon dioxide and water will combine to form carbonic acid in presence of enzyme known as carbonic anhydrase. And this carbonic acid will divide into bicarbonate, which is very essential pH buffer, and the proton. which is the like villain of the story. So this proton will be thrown out of the cell with the help of very strong pumps present on the luminal membrane of these cells known as proton ATPases, i.e. hydrogen ATPases, more preferably proton ATPases. These proton ATPases are so strong that even if there are 1,000 protons here and only 2 protons here, they will they will eventually cause these 2 protons to move out. They can work against the concentration gradient. They are so powerful, they do not mind the concentration gradient. They can push proton out against 1,000 concentration gradient. Okay, so what to do with this proton? This proton is like active, and it can attack the membranes, it can attack the cell. So we need to combine the proton with something. So which thing can combine with the proton? First of all, this is the dibasic phosphate and this is ammonia. These are like heroes of the story. The dibasic phosphate will combine with the proton to form H2PO4, and ammonia will combine with proton to form NH4, and chlorine is present inside the lumen, so it will combine with chloride to form NH4Cl, ammonium chloride. So let's discuss about this. First of all, there are 2 types of phosphates, monobasic and dibasic phosphate, H2PO3 and HPO4. They reaches the late distal tubule. Okay, from the GFR and all of the lumen stuff, they will reach the late distal tubule. They will combine with hydrogen or the proton to form H2PO4 and NH4Cl. So it means that it's a good thing. Second important thing worth mentioning here, that carbonic anhydrase, this enzyme is very sensitive to our body pH. It means that when our body pH is maintained about 7.34 to 7.45 to 7.35 to 7.45. I'm sorry for the misunderstanding. So our blood pH is maintained around about this 7.35 to 7.45. Carbonic anhydrase is a very important enzyme used to maintain our pH in case of acidosis. Okay, in case of acidosis, when our blood has a lot of acids like protons, it has a lot of lactic acid or sulfuric acid or etc. So carbonic anhydrase will start to make this bicarbonate. From where? Carbon dioxide and water will combine to form carbonic acid, and this carbonic acid will dissociate to form bicarbonate, and it is eventually released into the interstitium, and along the blood it can move into the our circulatory system. So it means that when a lot of bicarbonate is being made, what the other thing will be made more? The protons, because the carbonic acid is broken down equally into bicarbonate and proton. So what does it mean? It means the patient with acidosis will have more concentration of H positive in the urine. But as we have discussed that this proton is not alone, it will combine with either ammonia to form NH4Cl, or it will combine with dibasic phosphate to form H2PO4. It means that in the urine of such patients, we will find more concentration of H2PO4 and NH4Cl. Okay, it's like more conceptual thing. Also, one other thing it's worth mentioning that some amount of potassium can also be reabsorbed in the late distal tubule, and this late distal tubule, this potassium will combine with bicarbonate to form potassium bicarbonate. Okay, this bicarbonate is also not alone. Bicarb can be combined with potassium so to form potassium bicarbonate. It can act as the buffer in the circulatory system, and it can maintain the pH of our blood. This was for today. I know like some of the stuffs can be messing, but I hope you understand the video. In the next video, we'll discussing about the medullary collecting tubule, and till then, goodbye, take care.